Okay, so today we're going to be talking about a new workflow that we've added to our NGS tools. For quite a few years, we've had different workflows, RNA-Seq and ChIP-Seq, and SNP analysis, and short RNA analysis. And they would go through our workflows kind of in a linear fashion, and then at the end we could try to compare them. And what we found is that there were some great advantages to taking the data and combining them after the fact. And there's several different examples of when you may have data from different sources, but then you want to co-analyze them together. So we streamline that process tremendously, and it's going to be a pretty exciting webinar just to show how we can take very large data sets from different sources and do a combined analysis on them. And so we'll start today by some PowerPoint, and I'll introduce you to the company and some of the background for our NGS, and then we'll spend the majority of the webinar going through project setups and then some of the downstream analysis. And then, as Sharon mentioned, if you do have any questions, please chat them in, and I'll try to get to them as we proceed. So combined analysis is specific to NGS, but our software really is the DNASTAR software covers all different areas. And so if you're new to DNASTAR, please be aware that we have a full set of molecular biology tools, a full set of structural biology tools, as well as what we're going to talk about today, genomics tools. And so the molecular biology suite, I'll just give you just kind of a brief rundown of what we have here. These include all different tools like sequence editing, multiple sequence alignment. We have tools for Sanger data analysis. And so this is a set of software programs that has been available for, over 30 years at DNASTAR. So this is part of all of our packages. So if you have NGS needs plus some basic needs, we can cover all those different functionalities. We also have some newer software in our genomics suites that we're going to talk about today. The genomics suite, of course, includes Assemblies software, so it takes raw input data from sequencing instruments and aligns or de novo assembles it. And then we have all different types of downstream analysis tools depending on the different workflow that you have. We also have protein analysis tools. We call this our structural biology suite. And again, we have other webinars that focus in these areas, but we've done a lot of development lately in structural biology, and that includes some very nice interface for looking at 3D structures and predicting structures of proteins. So again, it's a comprehensive suite of tools, and we have fully functional demos for all these different tools. And we have a full set of webinars and videos that cover these in detail. So we're going to focus on the genomics suite today. And one of the first things I'd like to really reinforce is the idea that we've developed these tools to be compatible with all the different sequencing platforms and all the different NGS workflows. And of course, there's all different types of workflows, and ranging from de novo transcriptome to RNA-Seq to panels to genomes and multiple genome comparisons. And sometimes you want to do a little bit of each. You may want to look at, you've got a biological sample and you want to know, you want to know the gene expression, and you want to know the variations in that same sample. And so really the webinar today is focusing on how we can cross-compare these different workflows in a streamlined manner, in a more efficient manner. And the fact that we support multiple different sequencing platforms. Many of you are working with Illumina, but we also have Ion Torrent customers, long-read technology like PacBio, and of course we're looking ahead to the future for nanopore technology, and we will be compatible with nanopore when it hits the market. And so it really is kind of an all-in-one solution. If you're currently using multiple different software tools to accomplish, work with data from these different workflows, our software can handle it all in one interface and one streamlined workflow. One of the most common questions that comes up is, what type of a computer do you need to run these assemblies? And of course, these requirements change, as computers get faster and as data sets get bigger, these requirements change a little bit. But what we really strive for is that you don't have to have a supercomputer to handle even the largest data sets. And so These modest hardware requirements do have some specific requirements, though, and that's the type of computer for anything aligning to, say, a human genome. You want a 4-core computer at a minimum, and most of the i7 chips that we have now can handle, they'll have 4 cores and some virtual cores as well, so 8 cores or more. So having more cores in the computer will speed up a lot of the assemblies. The amount of RAM that you need varies between 16 and 32 gigabytes. The higher RAM requirements are usually projects that have deeper data. So RNA-Seq is a common example of where, depending on how the RNA was isolated, you may have extremely deep coverage on ribosomal genes or on highly expressed genes. So if you have those sorts of RNA data sets, you definitely want 32 gigabytes of RAM. If you have smaller data sets, 16, and for microbial data sets, sometimes even less than that. So your core computer, it's a budget computer, a $1,000 computer that you can get anywhere will meet these requirements. The one specific thing that we want is a scratch disk. Anytime we're doing an alignment to a eukaryotic genome, a lot of the data gets written to a disk, and it has to be a dedicated disk. So you want to just plug in an external drive through USB 3. That's more than adequate, and then you'll get great performance. And so, again, if you have questions about hardware, please don't hesitate. If you're looking at a demo, let us know. The data set that you have and the hardware that you have in place, if you'll have success, and we can discuss that with you. There's also some options to do assemblies up on the Amazon cloud. So if you don't have hardware present or available to you, you can have an option of running the assemblies up in the cloud. Cloud also has some other advantages. You can share data with collaborators at different locations, or you can use multiple virtual machines. If you want to do, 50 exomes overnight, you can do that on the cloud, and 50 different virtual computers will complete the job for you. So there's some specific instances where the cloud is a really nice solution. So one of the focuses for DNASTAR the past couple years has been the accuracy. And so for any of these NGS workflows, whether it's ChIP-Seq or RNA-Seq or targeted resequencing, you want to be assured that the assembly algorithm is the most accurate that you can possibly use. And we've gone through great lengths to really validate what our accuracy claims are. And so we've got white papers on our website. We use public data sets. We provide in our interface the ability to set up the project so it'll calculate the accuracy for you for your data sets. And we're very comfortable saying that we're greater than 99.7% accurate aligning Illumina data and finding variations that are present in these known samples. And so we have a number of these data sets, and I won't go through all of them, but it's important to note that we have these data sets that have that are validated human genomes and exomes. And in addition to the accuracy, we have great speed. So it's a combination of the speed and accuracy. And these are some new numbers that we've generated with our version 14 software that was released a couple weeks ago. And just to give you an idea, the assembly times down here at human exome, for instance, here I can do a highlighter here so you can see that. A human exome for Ion Torrent, about 4 hours. For human exomes for Illumina data, we're under an hour, so anywhere from 45 to 50-some minutes. And this is deep coverage, 80x coverage of the exome. The Ion Torrent is a really deep exome, 162x. And so roughly, if you're working with Illumina data, a little under an hour per exome. Smaller genomes, we count those in minutes. And things like RNA-Seq, about an hour and a half or so for RNA-Seq. So these are really fast assembly times, and we're still obtaining, really nice accuracy. And so here's kind of a comparison, and this is a little bit older data. It'll get refreshed with the version 14, but it's a comparison. Then it has both the accuracy and the time, and the timings are actually the older timings, so they're a little bit slower. But so this is a comparison of Sigmund engine to CLC QIAGEN's software, and you can see that the sensitivity, here, which is a measure of the accuracy, we're at about 99.7, and they're a full percentage point lower. And where it really makes a difference is the false negatives and the false positives. We have a much lower false positive detection rate than the CLC software, and a much lower false negative rate. So in this exome example, so this is a validated genome and exome data set, NA12878, we have a much, much lower false negative detection rate, and that gives us this better accuracy and a little bit faster. Now we're about another half hour faster for the elapsed time. So we think we're in a really good position in terms of where our assembly software is. And of course, it's the same software. You don't use a different algorithm between our targeted resequencing that produces these results and our RNA-Seq and ChIP-Seq. It's all the same algorithm. It works the same way. So you get the same benefits. You don't have to switch to a different algorithm for these different data sets that ultimately you may want to compare to each other. Another piece of relatively new piece of functionality is the human variant annotation database. And you'll see that when we set up our assembly projects that we're able to pull in database information. And from this variant annotation database, it comes from all different sources. There are functional predictions from SIFT and polyphenol mutation taster and conservation scores and ClinVar clinical information, and then also genotype and allele frequencies from 1,000 genomes, and of course dbSNP. We bundle it all together and automate just pulling this information into your analyses. And so the new software, this version 14, has streamlined these workflows so that we can do these different analyses and pull in database information. So the focus of today's webinar really is how do we combine the analysis from these different workflows? So we use the same underlying assembly algorithm, and now there's different ways that we can kind of merge the data. So there's actually 3 different kind of categories of combined analysis. So for example, if my workflow is RNA-Seq, with RNA-Seq data, in that data, there's SNP information, and I can also calculate RPKM gene expression values. And so those use 2 different types of kind of analyses, but we can combine them. In that data, we have that, that information. And so our latest software will run SNP analysis and gene expression and can do it on multiple different data sets. And so if I have multiple projects, I can analyze them across the projects for SNPs and for gene expression. And so one type of combining is just within a single data set. And you can see here for targeted resequencing, we can combine SNP calling and CNV. So we have SNP calls and CNV. And for genome, we can do the same thing. ChIP-Seq, we can do SNP calling and peak detection. miRNA, we can do multiple things, SNP calling, expression, and novel discovery or peak discovery. And so within a single workflow, we're going to combine analysis. We can also, a second type of combined is, for example, I may have 50 RNA-Seq assemblies that I ran last week, and then I generate 2 more this week. In order to compare all the RNA-Seqs together, the signal has to be normalized. You have to kind of reprocess the signal to normalize across all the samples. So a second type of combining is just multiple projects, same workflow. And so you don't have to reassemble all the different projects. We created a very streamlined workflow just to add them and recalculate the gene expression signal. So that's the second type. And a third type is combining data from different types, combining assemblies from different types of workflows. So for example, RNA-Seq and ChIP-Seq, so 2 different biological, it might be the same biological sample, but a different sequencing run, because you have different sequencing technologies and preps that generate the RNA-Seq and ChIP-Seq data. But I want to co-analyze them together. I want to relate my bound protein, maybe a transcription factor, to changes in gene expression. And so we've streamlined that as well, so we can load RNA-Seq data together, projects with ChIP-Seq, and then in the end get an easy-to-use interface for comparing the 2. So it's kind of 3 different ways that we can combine. It's all new to version 14. And that's something I can show you now in the software, how the interface has changed to accommodate this. And just one last slide. So kind of and we'll look at this in the software. So what we have here is an array star, and this is one of the tables where I have a SNP table, a peak table, a gene table, and a scatter plot, all in one view, all visible in a gene table. And now I can compare SNPs and peaks and gene expression, find interesting genes, and then visualize those in a new program. It's our GenVision Pro, which is a browser program that I can go from these gene tables and automatically go to that gene and visualize the different tracks of data. So it's a nice integrated. So that's really going to be the focus then, is showing the different ways that we can combine data, and then how we can do that combined analysis downstream, both in RNA-STAR and in our new GenVision Pro application. So Sigmund Engine is the starting point then for our NGS workflows. And typically we're going to assemble on our local computer. And so as I make selections in this wizard, you can see we get kind of a history of the pages that we've been to. And we choose workflows, and as we make our different choices, we kind of go down different paths. And it's really designed as a wizard to allow you to set up all different types of projects. But you don't have to have expert knowledge on bioinformatics to set up the different workflows. We try to design it to be as easy to use as is possible. And you'll see that as I pick different workflows, in our new software, this latest version, for example, if I pick whole genome, and I'm just going to pick a reference-based assembly normal workflow, and there's some new Sanger validation we'll cover on Friday. It's a new workflow for using Sanger and NGS data together. And we get some system information. And again, I referred to the technical requirements earlier. This is a 32 gig RAM machine. And what's very important is this temp file location. And on this computer, it's a dedicated E drive and a temp folder. You want to make sure that if you're doing a demo that you have this space. That's critical to success of your alignments. So we click next. We can add reference sequences. There's a new button here to download Ncbi genomes. So this is another little piece of functionality that can be really handy. And it connects to Ncbi, and I can pick the genome by accession numbers, or pick my domain and then browse through genomes and download them locally. So that's a nice handy tool. So it can be surprisingly difficult to find reference genomes at Ncbi. But if you're working with a main model organism, of course, we want to download a genome package that comes from the DNASTAR website. And so these are packages that we make for these model organisms that include the annotated reference genomes plus dbSNP. And so we combine them together so you have combined analysis. The human packages also have things like GERP, and they may have COSMIC depending on the version that you have. And so I'm just going to load in. I've done this already, so I'm going to add a genome package. And, oh, I didn't. So I have one here. So I can load that into my project. And click. I'm not going to load a VCF file here. So then I can load some data in. And we'll just grab any old data here. This is a human genome data set I've been working with lately. Of course, and I can group. So if you're doing, whether it's genome or RNA-Seq or exomes, I can group them and just name them. So this could be a sample 1. So I could set this up to do really as many as I want. So there's no real limit to how many we can do at one time. It'll run them consecutively. So we'll get sample 1 assembled into one group and sample 2 into another group. So you can set up all your assemblies here in this interface. And if we have a control, we can set a control. And this is the page that is has some of the combining. So again, back to the combined idea that we have these combined workflows. You can see now I can do a variant detection mode. I can say this is a diploid, and I can say calculate copy number variation. So for the targeted resequencing and for the whole genome, we can now have both type of analyses together at the end. And I can also import the variant annotation database. So this was previously downstream in RNA-STAR, and it took a fair amount of time to connect in with this database. Now we're going to tell it up front in our assembly that this is a human data set. We want to load in all the database information, and we want to have both copy number and SNP detection available to us. So that's an example of combining these two. Now if I go back, and you'll notice that as we pick different workflows, I go to transcriptome now, it changes a little bit. And so for transcriptomes, I don't get copy number variation. I'm not going to do that with an RNA-Seq, but I may want to do variant detection, or I might not. I might say I'm really not interested in SNPs with this workflow, but I can turn this on. And then I also have the ability to import the variant annotation database, and I can pick my normalization method for gene expression analysis. And so there's some advanced options as well for the alignment, but also for the variant detection. So depending on the workflow that I have the ability to combine analysis for a couple different things. And so each one of these workflows then has been updated to do that kind of a combination. So I'll just go to one more here. We'll go to ChIP-Seq. And ChIP-Seq is a little different. I can specify a control. Right, and now I have variant detection, and I have normalization and peak detection methods. Right, so I get SNP calling and peak calling together in one project. And when I go to the advanced options, I have some peak calling options here as well. And of course, these are all explained in detail in our help, which is contextual. So if you haven't seen these, you can go to help, and that'll explain what all the different advanced options are for that window. So that's the first type of combined, is within a single workflow, we can do additional analyses. Now I'm going to show you another. I'm going to go back here all the way to the start. And you can see this create combined project. So now I'll use the example. If I've got RNA-Seq projects, maybe I ran some last week and I have some new ones and I want to do a combined analysis at the end. I can select this new workflow. So what that assumes is I've gone through and done my RNA-Seq assemblies, and I have .assembly output files for each group. And now I want to take all of them together and look at them together and compare gene expression across all the samples. And so I choose this new workflow now, this new option. And what it's going to ask me is to add either the projects or the folders that contain the projects. So in this case, my projects are over here on my data drive under these ethnic exomes, and I've got some exome assemblies in one folder, and we can see there's 1, 2, 3, 4, 5, 6, 7 assemblies here, and then a latest folder where I've got 3 more assemblies, and I want to combine those into 10 different assemblies. So I can add folders and say, well, let's load this folder, and it'll look inside the folder. And what it's doing is it's finding those 7 assemblies, and it's looking inside of them, and it can say these are exomes, so these are DNA-Seq, right? So they'll have SNP information in them, and they may have copy number variation in them, CNV. So now I can add another folder, and so it went and loaded the remaining. So I can keep doing this so I may have 100 different exome assemblies that I want to compare. And so I can load those all in, and the software will look inside and tell me, what's present. If I have ChIP-Seq, I'll have chip controls. It'll recognize that these are ChIP-Seq and what the controls are. So it's got some smarts to it that it looks at the projects and determines what's in those projects. So now it's looking at the different data that's loaded, and it sees that we want to import the variant annotation database because we have the right human genome build. It'll do CNV calculation and calculate SNPs. So one possibility is that I didn't do CNV for one of the samples. If I didn't do that, it will recalculate it for those samples so I can compare them. And so depending on the different types of assemblies that I want to combine together, you can see I'll have different options available to me. Gene expression or peak or miRNA, gene discovery. And because I'm combining projects that have CNV, I have some control over how I want to compare to the different types of features that I want to do my copy number variation. So we have this area that's active down here. And now when I click next, I just call this combined exome, and what's going to happen at the end, I've already got the assemblies. I'm going to produce a .ASTAR project. and that is going to be a single ArrayStar project where all the signal has been normalized and put into one ArrayStar. So that's the goal. Previously, we had some of the capability within ArrayStar to import these assemblies. It was just far too cumbersome. So we designed this new wizard that just says, take all the assemblies, run them through our Sigmund engine, which has the very powerful and fast way to crunch through the data, and then give us a project at the end that's ready for analyses. So that's the second type of combination. Now I'm going to go back and show you a third type, and that's the one that we're going to actually look at the ArrayStar project and some of the output. So I hope I'm able to keep that clear. But so we'll go back here, assemble on our local computer, combine, and now I'm going to add some different folders here. So I'm going to go into this human transcriptome folder where I have some human RNA-Seq and some human ChIP-Seq. Oh, different folder I have to grab. So it gave me a warning there. This one. So that warning that came up said, wait a minute, the genome templates weren't the same. So that's an important point, is that if I have ran assemblies with HG18, maybe a couple of years ago, and then ran new assemblies with HG20 or HG19, you'll get a warning message. And when that happens, you have to reassemble and use the same genome template then when you do these kind of combinations. That's an important point. So these ChIP-Seqs then, there's a control and two different ChIP-Seq assemblies, and then the RNA-Seq in that folder was a time course. So it's a 0-time course, a 12-hour, and a 24-hour. And you can see that when I loaded these different types of data, now they come from different workflows, we recognize those and label them appropriately. And because there's ChIP-Seq, there's a control specified, and so it tells me what that control was called, and it verifies that my different assemblies contain that same control. So it's a check to make sure that we don't have different controls set up for the different ChIP-Seqs. So now when I go to the combination page, we have more things active. We have gene expression for the RNA-Seq, we have peak detection, and we have calculate SNPs. And one thing we could do is go back and say, well, I changed my mind. I want to do a different type of peak detection. And so I may have done MAX before, and I may say, well, I really want to do E-range 3 this time. And so another reason to do combined assemblies is maybe you want to get all the projects kind of together on the same page and say, I want to analyze them this way for SNPs or for peak detection, or I want to pick a different normalization method across all my samples without having to redo all my samples and reassemble all of them. So the combination allows us then to get all our projects uniform using the same kind of signal crunching here. So let me just show you one more thing. I'm going to go to advanced options. And so now I want to go to these advanced options. I can change for the peaks. I can also change in the detection options. And that's pretty common with peak detection, is that we may change the stringency to say, well, what I used before, I didn't find enough peaks, so maybe I want to change what the fold enrichment is or the number of reads. And so we can fiddle with that a little bit and see how that affects our signal. Okay, so once that's done, I just click Next and Next again. And what happens then is we get this script at the end. And the script at the end then is the instruction then to Sigmund engine to take all these projects and combine them and recalculate the signals. And it says start combining, and I'll just click it to see what happens here. And now we can see the progress. And what I always recommend to customers is, especially if you're new to the software, is that if you have any problems, if we start to see some red errors that come through here, you can export the log and send that to our tech support. You can also go back to the script. So when this is running, a script file is written, and so that will be in your output folder, which is over here. So these .script files are also invaluable. So you can send those and we can see exactly what you're trying to do. And usually between the script file and some knowledge of the hardware that you're using, we can troubleshoot virtually any problems that might come up. And so we can see here, it's just going right to QSeq, going through the data and calculating a new signal. And it's just going through the experiment. So it doesn't take that long. So it can go through these projects, it loads them up, makes a new signal, gives us analyses. So it's vastly faster than realigning all the data sets from scratch. So it's a huge advantage if you're, building projects up where you have a collection of assemblies and you want to start comparing them. It's a huge time saver to be able to combine them at the end. So we'll stop this right now. I don't know how much bandwidth this will take with our webinar, so I'm just going to quit this. And when it's done, I'll have the option here just to kind of open it right in ArrayStar. And that's typically the next place that we want to go is our ArrayStar software. And let me get this view how we want it here. Let's see if I'll try to dock it here. Come on. View. Okay. And so we'll start here with the experiment list. So when I have a combined project that has RNA-Seq and ChIP-Seq, and I've also calculated variations, my experiment list then will show me that I've got RNA-Seq, ChIP-Seq, and then variation information as a third type of experiment. And so ArrayStar now has been, with this kind of a redesign, is able to accommodate these different types of data that have different types of signals in a single project. So that's something that's new with this version of ArrayStar. Older versions could do it to an extent. This version really embraces the idea of having different data types and having the capability to compare them. You can see just what I have open here. I have a SNP table, I have a peak table, a gene table, scatter plot. So this latest version of ArrayStar can accommodate all the different data types and display them together. So the question is, for a data set like this, I probably want to look at gene expression and try to correlate upregulation or downregulation of gene expression with my ChIP-Seq results. And in this case, it's a transcription factor, and I want to see, is there a peak where there was a bound transcription factor near any of the genes that show upregulation, over this time course. So it's pretty common kind of analyses, and there's multiple different ways that we can do this analysis in ArrayStar. I like to just kind of show the simpler ways first. So one way is to use a scatter plot, and the scatter plot is, let me just expand this a bit here. There we go. And so the scatter plot that I have set up is showing 0 compared to 24 hours on gene expression. And so I can change this. At 12 hours, it's a little bit more compact. Some of the differences aren't showing quite as strongly. So I'm going to keep it at a 0 to 24-hour comparison. And what's nice about ArrayStar is that the different views then, there's some shortcuts to doing some filtering. So I can go dive right into the filter all that gives me a very sophisticated kind of a filtering where I can filter on genes, on fragments of protected areas that were covered, bound by protein, by peaks, by variations. So there's isoforms, exons. So I can search all different criteria and build sets, gene sets, and then compare those sets. And that's a common way to use ArrayStar. And we can see if I stay at genes, I have all different filtering criteria. And we've got multiple webinars where we really kind of dive into how to use the full capability of the filters in ArrayStar. But there's also quicker ways, and we create hyperlinks and ways to find what we want without having to dive so deeply into the filtering. So the scatter plot's a great spot, and you can see I can select in the scatter plot a range of genes. And in this case, I picked genes that had a relatively high, I didn't take all the genes that had this low level, but I just picked a swath of genes that show upregulation at 24 hours and meet a certain kind of threshold. They're above -2, log 2. So this is all on a log 2 scale. So here's all, a pretty good subset of about 2,000 genes that show some upregulation between 0 and 24 hours. And once I make that selection, you'll notice in the details panel on the right, I've got all sorts of options now of what I can do with this sub-selection. I can cluster them. I can just show them in a gene table. I can make a gene set out of them. So that's one thing that we might do. So I can select that, and this is now just the gene table of those upregulated genes. I can do things like manage these columns. I can throw a fold change in there. So here's a fold change column. I can manipulate these columns, and there's this new column manager that was new to 13. It's really, really nice. And because a gene table can get populated with a lot of different things, I may decide that I don't want everything that's there. So I could say, well, I don't right now want these peak values. So I can remove columns. I can change the order, and kind of get this table to look how I want it. So now I have some fold changes, and I have the log2 values then of my particular RNA-Seq RPKM values, and I also have peaks that are nearby. So that's one way. So I could do this and say, well, here's a gene set. So I'm just right-clicking here, and I can remember this as a gene set of upregulated genes. Of course, as we build sets, then I can compare those sets as well. But there's also action links. So I can select this region and say, well, I really want to compare genes to peaks. I want to see, are there any peaks near these genes? And so we've got these quick links that says, show the table of peaks that are near this subset of genes. And this will open a peak table now. It takes us a little bit for it to build the peak table. So I'll let that, usually takes about 15 seconds or so, so we'll let that build. So now I have different information. So this is all the peaks that are located near those upregulated genes. And so it's a nice way to kind of cross-compare then in this combined analysis. Of course, I could go and apply some filters for minimum values. This particular table, I'm sorting here on the RPM value for the peak. So I brought the bigger, stronger peaks to the top of my table. And now, at this point, I've got a subset of both downstream genes and intersecting. So I'm not quite sure where my transcription factor is going to modulate the transcription. It may be upstream in the promoter, or it might actually be within the gene itself, the gene annotation. And so, again, I have information then from these two different sources that gives me this kind of combination table. And now I can look at them. So I can see this top gene. I looked at a couple of these earlier. So here's one. I can select it. Again, I can populate it with more information, or I have a couple options. If I want to look at some of the peaks or the raw data, I can send this selection. One place I could go is Sigmund Pro. So, for example, if I'm looking at variations in peaks, I might want to look at the actual aligned data and see, look at the actual variant base. Or I might say, I really just want to look at the peaks between the different samples. Send this to the new program, and so in this case, we're going to send it to GenVision Pro. And so GenVision Pro is our new genome browser. This kind of first version is really designed to do just what we did here. It's designed to work within our software, not as much as a general purpose browser where you're just taking tracks in from all different outside sources, but it's really designed to work with RNA-STAR. And when I do this send, it's recording all the information that this is chromosome 17. It knows it's near this particular gene, and it's going to zoom in and automatically open the genome right in that place, hopefully with the right kind of zoom levels that we can kind of see the picture right away. And so it opens like this, and we can see GenVision Pro now. If I go up here at the top, there's a pull down for all the different chromosomes, so I can load in different chromosomes. And I can browse like a regular browser too. So it has this full kind of capability where I can zoom in, and look at different areas. And you can see as I move this slider around, I get the visualization and we have the chromosome view at the top, and at the bottom we've got our features, and we're zoomed in quite a ways. I'll zoom out here a little bit. So it's a real nice kind of a controller, so I can get the right zoom level on particular genes. And then we have the data tracks, which in this case are coverage tracks for, I have 0, 12-hour, 24-hour RNA-Seq, and then I have my ChIP-Seq control and the 2 ChIP-Seq experiments. And of course, I can control what's visible over here. There's a track viewer. So I might decide I don't need both the peak tracks, so I can turn those off. So I can control what's visible over here in the side panel, so I can make different coverage. So I might decide only to have 0 and 24 hours. And then I can also control what the peaks look like. And so when I start to make changes to the peaks themselves, to the tracks themselves, I have this options page, so I can do things like I can select them in mass. So I might say, well, I want to change the height of all of them to take up all the available real estate. And then I can start to do things that are, like change the color. Maybe my RNA-Seq, I want that to be, those tracks to look different than the ChIP-Seq tracks, so they pop out a little bit. So I've got some really nice controls here over, I can even change the kind of graph to give me a different type of graph. I can get rid of the grids, which I tend to do. And I can make my background, clear. So that I can change, and maybe I can make my ChIP-Seq different color, and even a different type of graph. And I can also change the scale. So you can see some of the peaks that are, these are in, this is a low signal here, so everything's on a 0 to 10. So I can change the scaling as well to be something that, so the goal here is to really make something that looks exactly how I want it. Now let's go back and find a more, a little bit more interesting gene. So I'm on chromosome 17 at this gene. A lot of times I work with a split monitor, so I'll just have these kind of two. So here's one that I can say, let's look at this gene. And it's designed to come up very, very quickly. So I've already got the plots up. So the interaction between RNA-STAR and the browser is almost instantaneous. It just loads the data. And here's, and it zooms in, hopefully, in this, to the right spot. So in this case, I've got my 0 hours now is moved. So I'm going to say, I want to look at my RNA-Seq. So I can move my tracks around as well. And I can see, oh, I have the color not quite how I want it. So I'm going to just reset. But this is zoomed in where I want it. Like that. So again, whatever your taste is. So you can move the tracks around and reposition them. And now what I can see is that the reason that we came to this spot was that we had this nice peak right on the 5' end of this gene. I get a nice plot of the peak, and I can see the control has nothing there, so it's almost no binding, binding in the control. And then this other peak for the 0 and 24, that's the RNA, that's the first exon. You can see we have coverage there, and I can scroll through now, and maybe I want to change the scale a little bit, so I can zoom out, and I see that nice strong peak. So that's a pretty interesting, when I'm comparing ChIP-Seq and RNA-Seq, that would be a pretty interesting position. And you can see that these exons, of course, are spread out. So at different scales, we see, the peaks look different. And another thing that I can do, I'll zoom back in a little bit, is change the scaling too. So the y-axis on these, it says, it's kind of small probably for you to see, but it says 0 to 60. Another common thing to do is to change the y-axis scale. So I might say, well, I want these peaks to be, a little stronger. So I can change that scale either one at a time, that just made it smaller, or I can say I want them to look bigger in my And then, of course, that was for one track, and I can do this for multiple tracks as well. Like that. So you can get just the right kind of level of zoom to get the image that you want, again, to do your analyses. So again, this is a new program, GenVision Pro, allows me to look at RNA-Seq data, ChIP-Seq data together. And the key functionality is that we combine the analysis from assemblies that we've run, bring them into ArrayStar, and then ArrayStar gives us the capability then to kind of cross-compare, to look at things like scatter plots, find gene subsets that are interesting, and from that, based on the expression pattern, to go and find protected fragments or peaks that correspond to the ChIP-Seq data. And of course, if I have other types of data, I could use, also go to things like a SNP table. And a SNP table I can filter as well. I could say filter all of them and filter at the variant level. And so I can do a variety of different filters. And here I'm just going to filter on these RNA-Seqs. And say, for example, choose SNP criteria. And I might say, just show me the SNPs that are really affecting proteins. And I can apply these filters. Some depth filters, some percent filters. And it will give me a list. In this case, there's 757 variants that could really disrupt a protein. And so from this, I can show these results in a variant table. And I can also say, show me, the genes that contain these variations. So you can kind of see, or I can remember as a variant set, but I can say, show me the subset of genes. So here's a subset of the genes, and again, I can remember these as a set. So I'm just kind of showing a little different way that I could use these. So I could say, remember these as a gene set, and I could label it, variant genes. And so the other kind of way to use ArrayStar is to make gene sets on different criteria, things like genes that were upregulated by a gene set created by expression, or a gene set created by variant analysis, or it could have a gene set created by where there's peaks. And once you start making gene sets, then you can compare them. And I don't know how well these correlate. I actually haven't done this exact filter. And I can do something like a Venn diagram and say, well, there's a subset here where the genes are upregulated and they also have a damaging SNP, which doesn't make, you probably want to see downregulated more, but I could go back and do different kinds of combinations then and then compare the different sets to look for sets that are unique to each group or common. And then again, I can, in a Venn diagram, select the intersection and do all different types of analysis just on that subset. So here's the subset of those genes that meet that criteria. And again, a little bit different way to use ArrayStar, but however you get your gene set, I can go here, and send that to GenVision Pro or SeqMan for visualization and further analyses. So with that, I would be glad to take any questions that anybody might have. And thank you again for spending the time with me today to look at some of these new capabilities in the DNASTAR software.
Thank you, Matt. We do have several questions that have come in from our attendees, and all three of the questions are with regard to the SeqMan engine part of your talk. The first question is there a limit to how many assemblies can be combined in the new workflow?
So the combination is, there really isn't a limit. We've tested it in terms of, a couple of hundred projects. So you could have a few hundred exomes or RNA-Seq samples. So it's got an extremely high capacity. So yeah, you can use it for pretty much to any scale that's reasonable.
Thank you. Next question is, can I combine assemblies if they were done using an older version of SeqMan engine?
That's a great question. So if you've got the software already and have been creating assemblies, you are able to combine, assemblies done in version 13, I believe version 12 as well, but older. Once you get to a certain point, the dot assemblies themselves do not contain some of the information that's actually needed to do the combination. But you'll be perfectly fine with version 13 and version 12. My recommendation, though, in general, is to reassemble, because there's always we continuously improve our SNP detection, our peak detection. Those get better with the later software. So if you're going to cross-compare, because the assembler is so fast, it's very easy just to reassemble. So my first advice would be to reassemble, but if it's relatively recent, version 13, then yeah, by all means, you can combine them.
And for our final question, I think you've already partially answered that with your last answer, and that is, can I combine assemblies that use different versions of the same genome, like different versions of the human genome?
No, it's not. It's not possible to combine those. So, if you're working in, again, whatever the genome is, you want to make sure that across the different projects that you have the same reference genome for each one in order to combine them.